Hang on one second. All right, welcome everyone to our Success with Microsampling in Clinical Trials live discussion, a Trajan Scientific and Medical and IQVIA Laboratories collaboration. I'm Libby Ellis, U.S. East Coast, sorry. I wish I were on the West Coast, huh, Tracy? U.S. East Coast Business Development Manager for the Neoterics Microsampling brand of Trajan Scientific and Medical, and your moderator. While I get started today introducing our wonderful uh, host, I'm going to just launch a little question here for everyone. Can you see my question?
Mm-hmm.
Yep. Okay, great. If you guys invite you to answer the question so that it can help us guide our discussion today. I'd like to introduce very pleased to introduce my Trajan colleague, Dr. Tracy Mizuno. She is our U.S. West Coast, Canada, and Australia Senior Business Development Manager. And we are joined by our friends from IQVIA, Decentralized Trial Solutions Manager, Dr. Julia Beck, and Decentralized Trials Director, Kelly Ofterheide. So thanks everyone for being here today. Really appreciate it. Tracy is actually going to lead us off today with an overview of microsampling, and then she's going to follow it with a case study and take a couple of questions afterwards. Then pass it along to Julia, who will also cover a case study followed by the best practices in microsampling. And then we're going to open up the floor for questions and hopefully some lively discussion. So, Tracy, I'm going to let you take it away.
Great. Thanks so much, Libby. And yeah, I'm seeing some of the answers to our poll question number one. It looks like a majority are currently exploring microsampling in clinical trials. So thank you so much for participating in the poll. Um, Welcome everyone. Thank you all for joining our Success with Microsampling in Clinical Trials. As Libby said, I am Tracy Mizuno. I am the Senior Business Development Manager at Trajan, and I'll be first going over a brief introduction of microsampling. So I think this would be really helpful for those that are new to microsampling. We have some folks joining us that are quite experienced. So yeah, it would be exciting to hear when we get to our live discussion, some of your questions and comments. So without further ado, let me go over what is microsampling, how it works, some of the key benefits, particularly in clinical studies, some technical or science behind consideration when when including microsampling in clinical trials, and some applications or assays that we've seen implemented in pharma clinical trials. So what is microsampling? Microsampling, first of all, is patient-centric, keeping the patient's needs at the forefront when deciding sample collection methods. So understanding your cohort, where the study is going to be conducted, things like that. And so some examples include home collection, maybe sending out a home nurse healthcare provider to collect to make it easy on the patient, self-collection, and including micro collections or microsamples. And so microsamples can help ease that sample collection, and typically it involves small volume and capillary whole blood, either liquid or dried blood samples. And it also includes self-collection, so or assisted. So in cases of a hybrid or a clinical trial or decentralized clinical trial, a caregiver or parent or guardian could be trained in helping collect a sample, or the end user alone can be trained to collect a sample whenever is convenient for them. So in addition to self-collection, that makes the shipping and handling more easier and procedures to ship out and lower the cost and burden on not only the site, but the end user. And that includes analytes that are stable at ambient. So either room temperature shipping or cold chain shipping is feasible with microsampling. So how does it work? So current methods either collect capillary liquid blood or capillary dry whole blood. The collection, shipping, and analysis of wet blood is typically achieved by uh, upper arm devices that where the lancet or microneedle is coupled with the collection container, or it could just be, a collection tube itself. Um, this allows for, again, shipping at either room temperature or cold chain shipping. And once it's at the lab, it can be further processed in the lab, either spun down, serum separated, et cetera, frozen, aliquoted. Whereas in sampling, microsampling in capillary whole blood that is dried, these are typically achieved by devices that are decoupled with a lancet or microneedle. And this typically allows for collection times to be less than 5 minutes, and devices such as volumetric absorptive microsampling devices can be utilized. For the collection. So then once it's shipped, minimal packaging or biohazardous treatment of the handling or of the package can be utilized. And then once it's in the lab, it can be directly eluted or stored for later on batch analysis. So whether it be microsampling with a liquid or dried capillary blood sample, deploying microsampling in decentralized remote sampling or patient-centric sampling, you're always keeping in mind the patient at the forefront, as I previously mentioned. And so this allows for low volume sampling, a few microliters, enhancing the patient experience because it is easily collected by the individual or assisted by a caregiver or parent. And this allows for faster recruitment because, again, this makes it easier for a sample to be taken at a convenient time, broadening the range of individuals that you'd like to recruit. So faster recruitment, increased retention and compliance in collecting certain data and assays that you would typically measure during your clinical trial. And altogether with that, as I previously mentioned, it could significantly reduce the cost of the shipping and handling, logistics, operations, those involved in at the site, and related costs in implementing microsampling in clinical studies. So once you've decided that you want to that microsampling will really benefit your clinical trial depending on your patient's needs, there's some technical considerations that IQVIA and Trajan we are happy to help support. We have experience in supporting our pharma clients deploy microsampling into their clinical trials, and so I kind of guide our clients, and I bucketize the kind of two technical considerations to keep in mind when thinking about utilizing microsampling. So that would be kind of the operations logistics side, which Julia will present a case study that focuses more on that. And then also the bioanalytical considerations or the science side of implementing microsampling. So as you see here, in terms of user technique, that would be standardizing the collection, understanding the stability of your analyte so that you know how to ship and store your samples during the trial. And then in terms of the bioanalytical side, we recommend to our clients to validate precision, accuracy, and sensitivity of their assay, whether that be a wet capillary whole blood or dry capillary whole blood, the extraction efficiency that's more specific to dried whole blood samples, absolute recovery and matrix effects, and most importantly, stability and aging of your analyte, so that you have a window of when to return the samples back to the BA lab. Hematocrit bias is specific again for your validation experiments for dried capillary whole blood samples, and utilizing devices like volumetric absorptive microsampling can help mitigate that across different hematocrit ranges. So now that we know how microsampling works and what it is, here are some examples or applications of assays that have been successfully implemented in clinical trials. So you can think of, your typical measurements in clinical trials, such as safety testing, pre-screening, PK, PDs, quite heavily utilized in microsampling, but as well as genomics, omics studies for biomarker analysis, in terms of application, microsampling has great utility when you're considering the cohort of your population, whether that be a rare disease or vulnerable populations like pediatric or neonates, persons with disability. This allows for inclusivity of a diverse population in your clinical trials. And lastly, monitoring. Your microsampling allows for frequent monitoring, and reducing the burden on the individual. So that is really seen in post-surveillance studies, phase 4, as well as kind of titering the medication that they take. So a companion diagnostic is also a great application for microsampling in pharma clinical trials. So with that, I'm going to now present our first case study, and it actually it focuses on, as I mentioned, the science behind the science and technical considerations when thinking about deploying microsampling in clinical studies. And this particular case study focuses on the population and kind of the key challenges of meeting multiple sampling that was required. So in this particular pediatric clinical trial, this was a phase 2 randomized crossover study that was conducted by PTC Therapeutics, and they published their results and findings in the Journal of Clinical Biochem in 2023. And the key challenge that they face in this particular rare disease population required frequent biomarker measurements in this subset, in this population, for not only diagnosis, but for disease status monitoring. So the current method for this particular biomarker is from plasma samples, and it requires a long analytical time and lacks specificity. So that was kind of the main challenge that they worked with. And obviously with having to collect large blood volumes at the clinic site from a trained professional, this brought on, quite a significant amount of anxiety and stress on the patient. And not only that, from operational side, the biomarker that they were measuring required a quick turnaround. So this overall was quite costly in terms of storage and cold chain delivery and shipping and handling. So they implemented a dried capillary whole blood collection, keeping in mind the patient and all these other parameters of their study. And they chose meter microsampling, a VAMS technology, volumetric absorptive microsampling, where they did a full validation of bioanalytical methods of both liquid plasma and a dried whole blood capillary. They were able to utilize VAMS because of its low blood volume. So they used the 20 microliter meter microsampling device, was able to reliably collect these samples by the end user or the patient, and they were reproducible and much better than the dry DBS card technology. So what they found in their in this particular study, their phase 2 trial, that the subjects, the patients, they were able to self-sample, but what they did find as they were conducting the study, that they had about a little less than 5% of samples that got returned to the lab that were incomplete in sample or a failed sample. So what they ended up doing was conducting a retraining, and that increased their quality of sample back to 100%. So all the samples that they received back into the lab with some appropriate training, that they were able to recover all those. And then, and in addition to that, they were able to collect at a convenient time and place. Typically, this was really important for the particular biomarker to be able to sample at a specific time. And in terms of the bioanalytical performance, they had a really great correlation to both the plasma and the Mitra dry blood methods with an R value of 0.976. And their stability analysis of the biomarker was assessed at room temperature and at higher higher temperatures as well. And so it was quite stable at room temperature or ambient, and even at stress tested, it was quite stable at up to 15 days. So this helps with the operation and the logistics of sending the samples back from either the home or the clinic site into the BA lab. And lastly, the accuracy, precision, and the hematocrit effect was assessed as well, and it was within 15% appropriate, which met the appropriate regulatory guidelines. And so the actionable outcomes of this study, which you can read, and if you want the publication, feel free to reach out to us, was that they were able to achieve a quick turnaround of the biomarker results, which was really critical for this particular study. It was convenient for the end user or the patient to self-sample, and it was the preferred method for their next study, which was a global phase 3 clinical trial that involved many, many countries and sites. And then just last July in 2025, they got FDA approval for their compound for the specific rare disease population. So this was a very successful example of microsampling in a pharma clinical trial. And with that, I'm going to pass it on to our friends at IQVIA. Julia will be presenting a case study that focuses more on the logistics and operational aspects and considerations when conducting microsampling in clinical trials. Stop here.
Thank you, Tracy.
Yeah.
All right, so I'll go ahead and let me share my screen for a moment, and we'll get started on talking about some of the other sides and other things to consider when implementing microsampling in your clinical trial. So hopefully you all can see my screen. All right. So as Tracy mentioned, her case study focused a lot on the scientific aspects of what needs to be thought through for implementing microsampling in a clinical trial. What I'm going to do is I'm going to talk about more of the operational and logistical considerations. So what we have here is this was a clinical trial that our lab ran, and it was an epilepsy clinical trial. And so the overall design of this study is shown here in gray. And what would occur is when a patient had a medical event, this would be defined as a type of seizure, they would have to take their medicine, and then within 30 minutes to 2 hours of taking their medicine, they would need to have a blood draw. That sample would then be sent to the lab for analysis. So the main challenge of this whole design is the unreliability of this medical event, which is shown here, because this is what jumpstarts pretty much all of the activity for this study. So in discussing this with the sponsor, we ran through 3 different operational types of options, and I have them listed here as option 1, 2, and 3. And I'll kind of go through each one and talk about the pros and cons for each. So this first one would be to domicile the patient. In this case, the patient would be in the hospital throughout the duration of the study. And the benefit of this is that there's really no risk of collecting that sample on time. However, as you can imagine, there's a lot of cons associated with this. First off, it's very inconvenient for the patient because they pretty much have to stay in the hospital the whole time. And also because of that, it's going to be very expensive. So this one was kind of set aside, and we decided to explore some of the other options. So in the second option, this would be another type of decentralized trial in which a home healthcare, either a nurse or a phlebotomist, would come and visit the home. So in this case, what would happen is following that medical event, which is shown here, and you can kind of see how we've got this timeline associated here. After that medical event, the patient or someone else in their home would call their nurse or phlebotomist, and then that nurse would come to that patient's home to collect the blood and to send that blood off to the lab. So for now, what's great because this patient is able to kind of live a normal life for the most part. They still have to be a little bit more aware, and they have to ensure that this nurse lives nearby so that they can get to that patient's home in time. But because of that, it is very costly because these nurses or phlebotomists, they will basically be on call 24/7 to assist with the blood collection. And even with them being on call, there still is a risk of getting the sample collected on time because, again, we only have about 2 hours following taking the drug to get the blood collected. So what the sponsor decided to do is to go with this third option, which I've shown here in green, and that is to do a self-collection. So in this case, what the patient was able to do is utilize one of our blood collection devices to collect dried blood. So again, as Tracy mentioned, dried blood has a much longer stability, which is another benefit of this study design. So what would happen is the patient, after the medical event, they would use that device either at home or if they're at work or they're on vacation. Again, we have a lot more flexibility here. And again, there's no risk for collecting that blood sample within the correct time point. And there's really not a lot of cons with this. There's not a huge cost increase. In fact, it's in some cases more cost effective. So this was the benefit of utilizing microsampling for this study. So let me now go into some of the case study findings from this. So I've kind of broken this down into 2 different buckets. The first one is thinking about the patient and how we can make this much easier for the patient. So the first one is just thinking about kit logistics. So for this study, the kits were picked up from the site during a previous visit. So again, they didn't have to go for an extra visit. This was all integrated within the study design itself. And then these kits were stored at home until they were needed. The other benefit is because, as I mentioned, this was a dried blood sample. These samples could be held at home and then brought to the investigator during their next visit. So again, trying to keep this as simple and easy for the patient as possible. Now shifting gears to talk about some of the other actionable outcomes. So these are things to consider if you all were to include microsampling in the trial, and that is to ensure that we have the proper temperature capabilities. So again, because these samples are going to be stored for a period of time, it is important to know, do they need to be stored at room temperature, humidity, that sort of thing. The other thing that we focused on is to prepare the patient in advance. That was done through site training, getting them familiar with how to utilize this device, and then also to provide detailed patient material. So instructions, there's also a lot of videos out there to show how to utilize these devices and how to store the samples. So again, trying to get these patients as familiar and comfortable as possible. The last thing is we also provided extra supplies both at the site and also for the patient to take home. In case something were to go wrong, there's no concern, there's no risk, and the patient can simply do another collection within the provided time scale that they need. So now that I've kind of walked through this case study, I'm now going to take a step back and talk about some of the best practices for if you all would want to implement microsampling in your clinical trial. And so I've broken this down into two different phases. So I'm going to first talk about the planning phase, and then I'll talk about what to think about when executing microsampling in a clinical trial. So for the planning phase, there are three main areas to focus on. The first one is the protocol design and consideration. So when writing a protocol that includes microsampling, it's really important to ensure that these remote visits are included early in the protocol development. And these remote visits should have a scaled down set of events. Again, for this type of study that I just walked through, the only event that really occurred was that blood collection. So we're not going to be doing a lot of other metrics, again, trying to make it easy for the patient and to allow for this study to continue. The other thing to consider is the patient population. So thinking about what type of patients would benefit from using microsampling. Now, there are a lot of patients who would benefit from this, but there are some in particular. For example, pediatrics, because we're collecting such a small volume, it helps them. Also, the elderly, we've found that this type of collection isn't very painful or nearly as painful as a traditional sample collection, so it helps with them. Also, mobily restricted and those who live in remote geographies, again, because now we're bringing this sample collection to their home rather than having to go to a clinic. And then lastly, the sample type. So again, there are two types of matrices that can be collected utilizing these devices, and that's either whole blood or dried blood. And Tracy kind of walked us through that, and I'm going to kind of recap that again. So here, if we look on the right, here's a summary of wet versus dried blood. So wet blood, these are best used for safety testing. So this would be a lipid panel or a liver panel, any type of chemistry panel. The other thing to note is that they do collect a smaller or a lower sample volume compared to venous blood. So they typically collect around 500 to 600 microliters of blood, but they also come in multiple sample matrices. So this includes SST, EDTA, as well as calcium. So you can do plasma or serum with this. And then also the other important piece is that stability and processing conditions must be established early. So this would include, can we delay centrifugation? If so, how long? What temperature do these samples need to be stored? Do they need to be frozen? Can they be shipped ambient? Those sorts of things will need to be assessed prior to including that in the study. And then lastly, dried blood. So dried blood is most often utilized for PK and PD type of testing. And the other important critical piece is that these bridging studies need to be conducted early on, very similar to talking about wet blood in terms of establishing the stability and processing conditions. And then the other piece is knowing how to elute these samples from that dried blood matrix. The benefit with dried blood, however, is that there's long-term stability and the logistics are a bit more streamlined because we don't have to consider centrifugation and typically don't have to consider cold or shipping type of conditions. So now that we've decided that microsampling is in your clinical trial, I'll go into how to execute this. So we've got a couple of different things to consider once microsampling is included in your trial and the trial is ready to go. So the first thing are the regulatory guidelines. So there are multiple devices out there. And so it's important to confirm that these devices are both validated and approved for the intended use in that regulatory area. So again, a lot of studies are conducted worldwide, and a lot of these devices have different approvals, both in the US or in Europe as well. So be sure to check that. The other thing is to iron out the shipping conditions and how these samples and kits are going to be sent both to the patient as well as to the lab. So the outbound components of, are these kits going to be picked up at the site, or can they be sent directly to the patient's home? So both of these options are possible, but it's best to discuss that early on to figure out what would be best for your trial. And then lastly, how will the sample be sent to the lab? Will the sample be dropped off at a courier, or will the courier pick up that sample? And there are pros and cons to each of those approaches. And then lastly, as I mentioned earlier, I'm talking about wet versus dry blood processing is another thing that needs to be confirmed. So for whole blood, the centrifugation and temperature conditions, and then for dry blood, of those elution requirements and temperature conditions will also need to be ironed out and confirmed that those can be followed through in the execution phase. So thank you all for your time. I'm now going to pull up our resources slide. Feel free to scan the QR codes that we have listed here. For IQVIA laboratories, feel free to reach out to either myself or to Kelly Ofterheide. Our email addresses are listed here. We also have the QR code for both our website and our LinkedIn page. And then if you would like to look at Neoterics by Trajan, we also have those QR codes listed as well. And as Tracy mentioned, they do have that publication regarding the case study that Tracy walked through. So please feel free to reach out to her if you would like to gain access to that paper. And then lastly, we also have another third-party resource that we'd like to include, and that is the Patient-Centric Sampling Interest Group. They are more of a nonprofit consulting group, and they've got a lot of other resources out there to help people include microsampling in their study. So the contact information is also shown down at the bottom left, as well as their website and LinkedIn page. All right, and thank you all so much. And now I'll hand it over to Libby to address any questions or poll questions.
Okay, thank you, Julia and Tracy. Great information. Appreciate you so much. I'm gonna throw up one more poll. Can you see my poll?
Yep.
Okay, so just a little more information to help us with some, discussions today. And while that poll is up, I wanted to let you know we'd like to open up the floor to all of you for asking any questions of any of our hosts today. You can either send me a chat directly and I can ask your question, or you can raise your hand and then I will unmute you so you can have a live discussion with any of the ladies. Did you guys see the poll results?
Yes. Yeah.
Okay. So I don't see any questions right now, but what we're going to do is, guess what? I have one more poll, and then we might ask you a question.
Yeah, it looks like majority is very likely that they will include microsampling. So very good.
All right. Let me stop that one. And I've got one more I'm going to launch. This was a good question. Can everyone see this question?
Yep, number 3.
Okay, excellent. Oh, interesting. Okay. Give it 1 second and then, yes.
Yeah.
So that's interesting. Protocol design, analyte validation, followed by operational logistics, and then stakeholder buy-in. Okay, that's interesting information. So I'm going to ask Julia, Kelly, and Tracy, based on this is a really interesting bit of information here. Yes, we will have the presentation available for you on demand. Ruth will send you a copy of it. Thanks for asking. So do you, any of the three of you, want to ask a question of our attendees about these three things that seem to be high on the list?
Yeah, so in terms of analytical validation and protocol design, the challenge there, yeah, I guess the question from me right off the bat is, specifically, is it, related to having the resources or having someone to speak to in terms of technical questions that you might have? Do you need to speak with, your preferred CRO, BA lab? Yeah, I'd be curious to know. Or is it the design, right? Like, how do you incorporate microsampling? Is it for PK measurements, a biomarker measurement? Yeah, I'd be kind of want to dig a little bit deeper into the majority of you guys answered that it was the protocol design and analytical validation that is quite challenging.
Does anyone have any input to provide?
I was going to say I can provide some feedback based on what we have seen from a lab perspective and through our work with Neil on the PC SIG is planning for implementing microsampling early enough in the protocol design process to be able to implement it on a study. If to realize the maximum benefits, I think it needs to be planned for in like the phase 1 trial design timeframe. And making sure that it gets implemented at that point will ensure that it can be utilized in like the phase 2 and phase 3 portions of the trial. I don't know if that is kind of the experience that was driving the answer to that question, but in my experience, that's what we have seen.
So you'd say that majority is done like the design is prior to drafting the protocol rather than there's an existing protocol and then an addendum is added to incorporate microsampling.
Yeah, because of the bridging and the validation that needs to go with it to ensure that you have correlation in a capillary sample, that has to be done early enough that it can be incorporated into the protocol. And that validation testing can sometimes take a while. And so I think it's making sure that you're planning far enough in advance for that.
Okay, thanks, Kelly and Tracy. And I do have a question. You guys, Ruth is asking, will trainings be available to clinical sites and Ruth, I'm going to assume you mean for microsampling in the clinical trials to have training available at those sites. So Tracy and Julia and Kelly, I'm going to let you address that.
Yeah, I'm happy to address that first and then, see if Julia and Kelly have any additional comments to add as well. So yes, Ruth, that's a great question. Is training available? That's something that Trajan we offer as a courtesy for pharma clinical studies. Our team is happy to support those training to healthcare providers. So oftentimes what we see is those clinic sites, we train those folks there, and then those individuals can either train the end user or they do an assisted collection. So we're basically training the trainees. And we develop a tailored presentation, work with the sponsor, work with the pharma company to ensure that additional notes or instructions are incorporated per your lab manual. And so, yeah, that's something that we are happy to support we find that it is really critical too for the success of microsampling. As I mentioned in the case study, They had a few, failed samples that were returned back. Failed meaning they weren't fully absorbed with the capillary blood. So there was some undersampling or oversampling that was observed, but very minimal. But again, these samples are very precious. So they wanted 100%, a good sample quality. And with appropriate training, again, they were able to achieve 100% sample utility. So they were able to process the sample for analysis. So yeah, Julia, Kelly, do you guys any other?
That's great.
Yeah, I think the other thing I would add also is that what we've done is when we include microsampling and we send out the patient instructions, we also provide a QR code that can be scanned and links to video training as well. And so that's another component that we have. So not only do they get the on-site training with that healthcare professional, but they also can take a look at that video and get virtual training as well.
That's great. Thanks. Thanks, Tracy and Julia. And I do want to point out, we're happy to have Neil Spooner with us from the Patient-Centric Sampling Group, and he's added a bit of info in the webinar chat here about web pages coming soon, it sounds like, Neil, for decision trees for clinical trials, and then he sent us a link for resources that might be of interest. So thank you so much for that. Hey, Neil. Thanks. Thank you.
No worries. It's a great webinar. Thank you so much.
Yes, appreciate you jumping in and sharing those resources. Fantastic. And we encourage all of you to visit the Patient-Centric Sampling website. Great information, and it is a way to expand the microsampling community. So we are very much in support of that. Okay, guess what? I have one last poll. Let's do it. All right, let me know when you can see it. Yes?
Yep, it's up.
Okay, great.
Oh. Okay.
Interesting. Okay, look at that, 50-50. No,
Yeah.
no, if you have other, you can certainly add it into the chat.
Yeah.
All right, so we're going to go 50-50 with that one.
Interesting. Okay. Yeah, I'll add, for those assays, both wet or dry microsampling devices are a great fit for both those applications.
All right. We have a few more minutes here before we finish up. Happy to answer any other questions that you have, or, via chat or raise your hand. I'm going to ask Julia, do you mind sharing your last slide before we finish up today? Just the resource slide, because it does have contact information, and really would be interested in any other questions you can send us via email, and happy to connect with any of you for any discussions. You just reach out to any of us on the screen here, including Neil, I would assume. So if you want to raise your hand and ask a question, we've got about 3-4 minutes left. And if not, thank you all for joining us. Thank our wonderful host today for all of your super information, and learned a lot, and for being available to have more in-depth discussions and conversations going forward with anyone who would like to reach out. And we do at Trajan, and I'm going to go out on a limb and say at IQVIA Laboratories as well, have colleagues around the globally that we can put you in touch with to have some conversations about microsampling success in your clinical trials. So we hope to work with you soon on some of your projects. Thank you so much for attending. We really appreciate it. And Ruth, thank you for asking great questions. Have a wonderful day.
Thank you.
Thanks, everyone.
Thank you.